This conference will now be recorded.
Good evening, students. I'm Dr. Divya. So, in the fifth chapter, Principles of Inheritance and Variation. Today, we are going to learn about gene interaction. Okay. So, this is very important topic, especially for your NEET exam. Okay. For board exam also, you will get questions, but mainly you'll get more questions in the NEET exam from this genetics part. And that about the ratios and all type of combination, different genetic effects and all, you will get questions. You will get more questions. So you should be clear about this part, the theory part and different ratios. So first, let me explain what is gene interaction. Okay. So up to this, we learned about the concept of expression of genes. The expression of genes, single or each gene is having their own expression, or that is having an individual expression, right? Each gene that will express. That expression, that phenotypic expression. Each gene is having their own expression, or that is having an individual expression, right? One gene, one phenotype. That concept only we learned till now. But there are some other type of expression also, okay, which is different from this. So the interaction between genes or genetic interaction, that's what we are going to learn now. So that is the functional relationship between genes. What is genetic interaction? It is the functional relationship between genes. It's the functional relation. So how one gene functions, that relation. See, one gene, both genes are interacting. Here we explained about one gene, how the expression is working out, or how a gene is expressing the phenotype. Okay, the phenotypic expression of one gene we learned till now. Here, genetic interaction between gene for the phenotypic expression. So how the function of some genes that results in the expression of a particular phenotype. That's what we are going to learn now. Gene A and gene B both will work together and it will form a phenotype. So that's what, that kind of interaction between genes, that's what we are going to learn today. So that is a functional relationship between genes. So here, what you are going to learn here from one gene, one expression, that is the Mendelian concept, right? That is the concept of Mendel's concept. Okay, there you got F1 one ratio, in F2 another ratio, and the phenotypic ratio and genotypic ratio, everything you got, right? So here the genetic interactions, that is the extension of Mendelian genetics. Here you will get deviations of that Mendelian ratio. So genetic interaction occurs when the interaction between two or more genes that will result in a phenotype, and that is different from the expected phenotype, right? See, one gene here we can tell that A, this was, and it is forming like red flower, and here this is forming white flower. But when these both interacts together, that is forming another color, like pink color. Just for an example, I'm saying. So this is here some interaction is taking place between these two genes. If there is no interaction, there is an individual expression, it will express as such. A will form red color, B will form white color. But when it is combining, it's forming another, it is giving another expression. It is neither forming the red color flower or white color flower, nor the white color. So there is a different expression. How it is coming, that different expression or different phenotypic expression? It is due to the interaction between genes. So these both genes are interacting, and this interaction that results in different phenotype that is not expected. Usually, according to Mendel's experiment, what we will expect here in F1, any one parental character, right? Here either it should be red or white. But here in gene interaction, that forms a different and unexpected phenotype. We don't know the phenotype of that individual. Okay, like that unexpected result it will give, or that is giving a phenotype that is different from the expected phenotype. Mendel, according to Mendel's work, we will expect a parental character here, but it is different from the parental character, or it is not an expected result. Okay, so I will explain in that a better way. For example, here we can take humans. In human being, we have several genes, right? Humans, they have several genes. Many genes we have. In this, all genes won't give a particular expression. Like here, thousands and thousands of genes are present. Okay. So each one won't form one character. Okay, this will form another character, this will form another. It's not like that. Sometimes the combined action, some genes interact with each other. Maybe this gene A, here I said the gene A and gene B, both will combine and it will form a character Z. Okay, so these group of genes interact with each other, and as a result of that group work, you will get, as a result of this group work, this kind of group work, as a result of that, particular expressions you will get. Okay, like skin color, eye color, hair color, all these are not a single gene effect. All these are controlled by several genes. It's a group work. It's a combined effect of many genes. Okay. So a single gene is not giving a particular result. It's forming different. It is a result of different genes and combined work of different genes. Okay. So some genes interact with each other, or a group of genes interact with each other, and as a result of this interaction, that group forms a particular expression. So in this case, we can't find a single cause, like a single reason we can't say for a particular phenotypic expression. Okay, so we can't say a single reason, like there is a phenotypic expression and behind that we can't say a single reason, because it is a combined effect or combined interaction of so many genes, and finally it is producing a product or a phenotypic expression. So instead, the genetic variation may combine with one another, right? There is a combination of different variations or different genetic reasons, and each variation that will contribute separately. There is a separate contribution of each gene, and finally the combined form of all these results that will form a specific phenotype. See, for example, there is a gene A, gene B, gene C, gene D. Sometimes this A gene that will work first and it will reach the C. And C, from the C, after the product, here some product will form, right? Some enzyme, something will form. This enzyme will activate gene B. See, if there is no gene C product, this gene B may not work. That cases also will be there. Okay, that I will explain. Each example you will get, that time you will be clear. So here, without this gene C, the gene B may not be working. Okay, and this gene C will activate this gene B, and this gene B will go to gene B. And after these combined effect of all these three genes, this gene D can give a product or a phenotypic expression. Okay, so this is a combined work. So this type of, here we can't tell that gene D is not giving a proper product, means it's not the fault of gene D. Here so many works are involved. Okay, so we have to find out in which point it is getting an issue. Okay, so the genetic interaction, it's like a network action. Okay, it's like a network action, genetic interaction. So the genetic interaction provides a clear idea about how genotypes connected to phenotype. From here, from this example, you can get that how a genetic interaction that is giving a particular phenotype, or how the genotypic condition that is connected to the phenotype or the phenotypic expression of an organism. So the genetic interaction studies that were done in many organisms. For example, all these genetic interactions, several genes are interacting. That studies were conducted in E. coli, Escherichia coli, then Drosophila, that in that so many genetical studies are going on, still it's going on, and several studies they conducted in Drosophila. Then Saccharomyces. Okay, Saccharomyces. This is a particular type of a species of yeast. Okay, so these are mainly studies, all these genetic interaction studies they conducted in these organisms, and that Mendel and other workers, they thought that, or they assumed that these characters are governed by single gene. Okay, that's why we said one gene, one phenotype. That was the concept earlier. During Mendel's period, that was the concept. Okay. So later they studied more and they conducted more and more experiments, and they proved that many characters, so many characters are governed by two or more genes. Okay. It's not one gene or two genes. Sometimes many genes will be involved in that. So many characters like that, those are governed by two or more genes. So that's how genetic interaction. Okay. So that made modifications in the Mendelian ratios or the inheritance of character. We thought that one gene, or in a monohybrid cross, we learned how characters are getting inherited. So we are expecting a result in that way. But later, more studies were conducted in this area, and they proved that there are many genes for one character. Many genes are involved in the expression of one character. There may be or may not be. Sometimes one gene will be controlling, but in most of the cases, many genes are governed by these characters are governed by two or more gene interaction. So that made modifications in the ratios, in the Mendelian ratios, okay, or Mendelian inheritance. So in gene interaction, expression of one gene that depends on the expression of another gene. Expression. Here I said a gene, that expression, this will form some character like this shape. Imagine. So this shape is not only with the effect of this gene. There are some other genes also, like B plus C, that works for A. Then finally A forms this shape. Like that, chances are there. Okay. So there are many genes are interacting and they are forming a final product. Got it? So the gene interaction means that gives an idea about how genotype is connected to the phenotypic expression of an organism, and the genetic interaction studies They were done in which and all organism? E. coli, Drosophila, then Saccharomyces. All these organisms, they conducted the genetic studies. And later, more and more studies they conducted, and later they proved that two or more genes are involved in the expression of a phenotypic character. In majority of cases, more than two genes are involved and their interaction makes a particular phenotypic expression. So there comes the difference from the Mendelian ratio. So these studies proved that there are deviations from the Mendelian ratio. Mendelian ratios, it's just like, it's not like one gene, one expression. It's like many genes are involved and there are some kind of deviations or difference from the Mendelian ratio also. So in a genetic interaction or genetic interaction expression of one gene, it depends upon the expression of another gene also. Okay. And here, the genetic interactions, we said, what is genetic interaction? We know that interaction of genes. Okay, these genetic interactions, they are of two types. Which are they? First one is allelic gene interaction. Next is non-allelic. Okay, first one is allelic gene interaction and next is non-allelic gene interaction. So genetic interactions are of two types, allelic gene interaction and second one is non-allelic gene interaction. So first one is allelic gene interaction. Here, what do you mean by allelic gene interaction? This allelic gene interaction is a kind of gene interaction that taking place between the alleles. Okay. Allelic, from the term itself, from here itself, we can make out it is allelic genetic interaction, means between alleles. Okay, between alleles of a single gene. We know that a gene is having different forms. Two different forms, they are called as alleles. Okay, so this allelic genetic interaction, this is between alleles. This genetic interaction is between alleles. Alleles are interacting and it is forming an expression. And it is forming expression or phenotype. That interaction results in a phenotype. Okay. So it is a kind of genetic interaction or gene interaction between the alleles of single gene. Okay. So this is called as allelic or intra-allelic interaction. Intra-allelic. It's otherwise called as, or it is called as intra-allelic. Intra means within that. You have learned about intracellular, extracellular. Intra means within that. Intracellular means within that cell. So here intra-allelic means within that alleles only. Okay. So in genetics, mainly you have to learn the term, these terms. From that itself, you will get half of the explanation. Okay. So here allelic genetic interaction or intra-allelic genetic interactions. Intra-allelic means within those alleles only all these interactions are taking place. And see here between alleles it is taking place, the interactions. Then it is forming an expression or a phenotype. Okay. So this is happening in between alleles. In between alleles of the same gene. So it's not between two different genes. Okay, it's intra-allelic. So within the alleles of same gene. It's not between other genes. Okay, it's between the alleles of same gene. So under this, which and all will come? Within that alleles, what and all interactions we studied? Let's see, we know that the normal monohybrid ratio. According to Mendel, monohybrid ratio. We have learned all these things. Monohybrid ratio, phenotypic ratio is 3:1. And the genotypic ratio is 1:2:1. Right? So we have already discussed about incomplete dominance, then codominance. Right? Incomplete dominance, then codominance. Incomplete dominance and codominance, both we have learned. So how I'm going to connect these two with these allelic genetic interaction? Do you have any idea? Like these are all coming under allelic gene interaction. Why all these are coming under allelic gene interaction? Because all these explanations we gave between that alleles, right? For example, in these cases, the interaction happens between alleles of same genes, right? We explained about ABO group, ABO blood group, that is the same gene, I gene, but it is having different alleles, IA, ID, and small I. These are the one gene, but different alleles. So within the same gene, different alleles. That is the concept, right? So in incomplete dominance, a dominant allele that is not expressing, it's not completely suppressing other allele, right? I'll explain. So we'll get more clarity about this concept. Intra-allele means between those alleles, right? So there is no other gene. From the same gene, you are getting all this. There is no external genetic interaction. So here, in incomplete dominance, let us take that example. Incomplete dominance. We have learned about incomplete dominance. So in this, one allele is not completely suppressing other allele. Incomplete dominance. The dominance is incomplete here. That means one allele is not suppressing. It's not completely suppressing. Not suppressing completely another allele. Right? So the heterozygous expression. Here heterozygous condition. This is the heterozygous condition. Okay, both alleles will come. For one gene, both alleles will be there. So the heterozygous expression is an intermediate phenotype. Right? This we have already explained that will form an intermediate phenotype. So that is different from the homozygous parental genotype. Right? So that's why in the Snapdragon, we have discussed about Snapdragon and the cross between pure red. Here, Snapdragon, pure red and pure white. This we discussed. So here, what you will get, it will form this condition. Here, red and this is white. But in F1, you will get pink flower. Okay, in F1. So that is the intermediate expression. That forms an intermediate expression. And here, this, it is due to intragenic interaction. Here, this is giving here pure red parent is crossed with pure white parent. But in F1, you are getting pink flower. How you are getting pink flower? Because here these genes are here the alleles of these genes are interacting together, and that forms an intermediate expression here. Due to that interaction between the alleles, here the dominant allele forms the red color, recessive allele forms the white color. So when these both alleles of the same gene, they are interacting together, and that forms an intermediate expression, and that expression is not exactly the parental character. That forms an intermediate expression of both the parents. Here it is forming pink color flower. It's not white color and it's not red color. When we are mixing white and red, we'll get, if you are mixing white color and red color paint, we'll get pink color paint, right? It depends upon the percentage of combination. So, like that, both genes or both alleles of same gene will interact and it will form a intermediate expression, which is not exactly like the parental character. So, this is due to intra-allelic interaction. Okay, within that alleles, all this interaction is taking place. Intra, the word meaning is within the gene. Intragenic means within the gene. That means the allelic interaction is taking place. So in the case of codominance also, here incomplete dominance, hope you got the point. Next is codominance. Codominance. So in the case of codominance also, we took an example of ABO blood group, right? ABO blood group of humans. So this is the interaction. Which gene is controlling the blood group or that blood sugar which is present on the surface of RBC? I gene. I gene is controlling. So this I gene is having different alleles. That is IA, IB, and there is a recessive allele, small I. Right? So this determines different combinations of this that forms the blood groups, different blood groups. So that here all in these example also there is an interaction when that AB blood group comes. There is an interaction between IA and IB allele, right? And that forms AB blood group. Here when IA and the recessive allele comes, here also interaction is taking place and it forms A group. Right. So all these, the codominance, incomplete dominance, how it should express or how the expression should be, that is determined by the interaction between alleles. So within the gene, the interaction is taking place and it is coming to a decision, a final decision, this interaction. See, when some people are discussing, finally they will reach in some decision or finally they will take some decision and there will be an outcome for that discussion, right? Like that here, alleles of gene, they are interacting together and finally a product or an expression or phenotypic expression will be forming. That is the interaction between alleles. Okay. Next is non-allelic genetic interaction, or otherwise it is called as intragenic gene interaction. Non-allelic. Non-allelic means from that itself it's clear that it's not between alleles. Non-allelic, it's not between alleles. Then where it is taking place? Otherwise it is called as, this non-allelic genetic interaction is otherwise called as intergenic. 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 Inter means between. Between genes. Okay. This intergenic genetic interaction is taking place in between genes. Okay. So it is happening in between two genes. Here, two different genes are interacting. Okay. These are not between alleles. It is between two genes. Here, two different genes are interacting and these are non-allelic. Right? That means it's not between alleles. Here it is not between alleles and it's between genes. So these are the interaction between genes which are located at different loci. It may not be in the same locus. Okay. Sometimes it may not be on the same locus or on the same chromosome or on different chromosomes. There are several chances. It may not be on same chromosome. It may not be on same loci or that may be on different chromosome. All these interaction will take place. Okay. So due to this non-allelic genetic interaction, each type of interaction gives different ratio. Okay. Between, see, may not be from same locus, this interaction. One gene of one locus and this gene of another locus that may interact together and that forms a phenotypic expression. That also can happen. And on different chromosomes, genes of different chromosome may interact. That is also one chance. And on same chromosome, interaction may take place, but it's not between alleles. It is between different genes. Different genes of different location can interact, different chromosome, different genes of different chromosome can interact. So all these chances are there. So all these interactions may result different ratios. So this results in the deviation of Mendelian ratio. Ratios that forms deviations from normal Mendelian ratio. So some examples we can learn. So all these are very important for NEET exam. That's why repeatedly I'm saying note down all these points. It's important for NEET exam. Okay. So among few examples of this non-allelic genetic interaction, first we are going to learn about complementary genes. Okay. Complementary genes. How that will complement? They are complementing each other. So how? We are going to learn that. Complementary genes, they are two genes. Okay. Complementary genes, the genes which are complementing each other. We said these are genetic interaction, gene interactions. Okay. This is non-allelic gene interaction. Means it is between different genes. Two genes are interacting, not two alleles. So here complementary genes, two genes will be present which are complementing each other. These genes are complementing. So complementary genes are two genes and these genes will be present on separate locus. Okay. Both genes will be present on separate locus and these two genes interact each other and they work together. They will interact, they will work together and they will produce a dominant phenotypic trait. Okay. The important point is if these genes present alone, they can't give that phenotypic expression. Okay. Here, the complementary gene interaction, how it will work? Two genes from different locus and these two genes will interact each other. They will interact. Two genes will interact Then they will work and they will produce a phenotype. As a result of that interaction, they produces a particular phenotype, okay, or a phenotypic trait. And what is important in this complementary gene interaction? The important point is that these genes, if they are present alone, see here gene A and gene B, this will work together and they can give a phenotypic expression. Okay, but A gene is present. A plus B gives a phenotype. Here A gene is present and here B is not there means no result. Okay, there is no phenotypic expression. That means they are working together or they are complementing together to produce an expression or a phenotype. Got it? They are working together and they are forming a phenotype. Here you can see the example of purple color flower of sweet pea. Okay, this is purple color flower of sweet pea. This is the complementary gene interaction example. So, purple color flower of sweet pea or sweet pea. The scientific name is Lathyrus odoratus. Lathyrus odoratus. This is the scientific name of sweet pea. Okay, sweet pea or Lathyrus odoratus. So this experiment was done by Bateson and Punnett. Bateson and Punnett. Note down the scientist's name, Lathyrus odoratus, in that plant, which is sweet pea. Sweet pea is common name and the scientific name is Lathyrus odoratus. The complementary gene experiment. This is an example for complementary gene interaction. In that purple color flower, the flower color, that is an example for complementary gene interaction. Okay. So this studies that was conducted by Bateson and Punnett. So they demonstrated that in sweet pea, purple color flower, this purple color flower, that is a result of two dominant genes. Here you can see the genetic or the genotype CC. Here the C is dominant. Here the P is dominant. Okay. Here two dominant genes C and P. That forms white color flower. Okay. When these dominant gene C and P, these dominant genes C and P, when these are absent, that produces white color flower. Okay. Here in the picture you can see these two flowers. We have mentioned the genotypes here. Capital C, small p, small p. Here in this white color flower, C is the dominant gene. Okay. And here also you can see P, dominant gene P is present. But here only one gene. I said capital C, this dominant gene C and P. When it's coming together, that forms a purple color flower. Okay. When it is coming in dominant condition together, that is forming purple color flower. Here in this white varieties, these you can see white varieties, capital C, and the P gene is in recessive condition here. Okay. Recessive allele is present here. So, this forms white color. And here, the C gene, the recessive allele is present, but P, dominant allele is present. Then also it will form white color flower. But when these are crossing together, both dominant alleles will come. Here C dominant allele is there, P also. Here this is the heterozygous condition. In heterozygous condition, right? One, each gene, they are having one dominant allele in the F1, right? So both dominant alleles are coming together. So these alleles, these dominant alleles are coming together and that forms purple color flower. Okay, when it is alone, it is not forming purple color flower. You can see here also this is dominant, but that another gene is not together, right? These genes are not together. When this C and P dominant genes are coming together, that forms purple color flower. Otherwise, it's forming white color flowers only. So here, these two flowers, we have mentioned the genotypes here. In this, you can see both of them don't have these genes together, right? C and P genes, they don't have together. It's single. Here one gene is there, here another gene is there. And in this, here, only one gene is dominant in these both flowers, right? So when either C or P in this dominant condition, that flower becomes white. Okay, here C is dominant, P is recessive. So the white color flower is there. Here C is recessive, P is dominant. Then also it forms a white color flower. When this genotypes white, this white and which one? Another white with another dominant allele. These both crosses, it forms F1 in heterozygous condition. But in heterozygous condition, both alleles are coming together. The capital C and capital P are coming in that. So both C and P dominant alleles are coming here together. So that forms purple color flower. Okay. Then from the Punnett square, you can see in F2 again this purple color allowed for cell fertilization. So in cell fertilization, after that it will form different gametes. You can see here these are different gametes, male and female gametes. When they allow for cell fertilization, that forms here. Already we said both dominant alleles are present means that forms the purple color flower. So here you can see in the first combination you can see both dominant alleles are present. So that forms purple. Here also both dominant alleles are present. This is also purple. Here also both dominant. Here also both dominant. This one and this one you can see. And here in this combination both dominant C and P. But here comes the difference. Here again only capital C is there. P is in the P gene is in recessive condition. So it will give only white color flower. Both genes should come or both alleles should be in dominant condition. Then only it will give purple color flower. Right? So this is white. And again here both alleles C and P alleles are dominant. It will give purple. Again here C. Only one C allele is dominant, so it's white. Here C and P both dominant, purple. Here C and P both are having single dominant alleles. In heterozygous condition both one, but it is having single allele, dominant allele. So that is also giving purple. And remaining this, here only one is having dominant allele. Here also one is having dominant allele. Here both are in recessive condition. Then also that gives white color. But it is in homozygous recessive condition. Okay. Both genes are in recessive condition. Homozygous recessive. Still that is also giving white color flower because there is no presence of dominant allele. So that won't give any other color. And here white. Here also only one dominant allele is there. And again here C allele only dominant. So all these are giving white color. 1, 2, 3, 4, 5, 6, 7. 7 white color flowers and another. How many are there? 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 purple color flower. So normally, in the purple color, normally what is the Mendelian ratio? F2 ratio 9:3:1. Right? So all these, due to that single dominant allele, these formed white color flower. Right? Here you can see only single allele is present. So those 7 formed white and another 9, remaining 9 formed purple color because in that combination they have both dominant alleles. So that gives a purple color flower. Got it? So the purple color that is given by the pigment. Here I'll explain how it is coming. In this plant, purple color, purple flower, it is the result of anthocyanin pigment. The pigment anthocyanin. The pigment anthocyanin forms this purple color. Okay. So here substrate A. Substrate A forms substrate B. Okay. So with the help of one gene, which gene? Which gene is working? Gene C. With the help of gene C, substrate A forms substrate B. And here gene C is working. Okay. So this substrate B, again the substrate B is forming anthocyanin. Anthocyanin. And here gene P is working. Okay. Here the gene C and P both are interacting together. And first from A, substrate B is forming with the help of C gene. Then from B, from this B substrate, anthocyanin. Anthocyanin is forming with the help of gene B. Here these genes are interacting. Here first interaction from this to this product, from substrate A to substrate B with the help of C gene. Then this B product is forming anthocyanin with the help of another gene. Okay. When these both are in dominant condition, then only this product anthocyanin will form and it will come as a purple expression, purple color flower. Okay. If any one gene is absent, this dominant condition is absent, or this C gene is not present in dominant condition, it will not result in the purple color. Okay. So here in Punnett square, white color genotypes you can see 1, 2, 3, 4, 5, 6, 7. 7 are in white color because both alleles, dominant alleles are not present in this, the C and P in dominant condition. So that is forming white color. When C and P both are present in dominant condition, C gene and P gene alleles are there. At least one dominant allele is present from both. It is forming purple color. So that is the interaction between those two genes. Okay. So from the checkerboard, it's clear that in F2, normal Mendelian ratio, we know 9:3:3:1. But in complementary genes, that ratio is modifying into, or it is getting modified into 9:7. So what is the ratio of complementary gene interaction? 9:7. These ratios you have to know. 9:7. And how it is? Both genes are complementing together and it is forming a particular phenotype, or it is forming a particular expression. In the absence of one gene, it will not give the complementary gene effect. Okay, because there should be another gene to complement this result. So that is a complementary action. And what is the result? The ratio means Mendelian ratio, it is getting modified and it is forming 9:7 ratio. Got it? Next example for non-allelic gene interaction is duplicate gene interaction. Okay, duplicate gene interaction. So in duplicate gene interaction, dominant allele of both genes produces same phenotype. Okay. In duplicate genes, dominant allele of both Both genes. It's interaction between genes. Okay. So both gene dominant allele condition that forms same phenotype. That forms same phenotype. But there is no cumulative effect. Okay. What I mean by that? Means it is not necessary to present together in dominant condition. In the previous example, what we came to know, in that what we learned, both should be present together. Then only it will give a purple color flower in the previous one. Right? In complementary one, both should be in dominant condition. Then only it will give a result. But here in the duplicate gene, it's not necessary to present together in dominant condition. If both present together or both present separately, that forms same dominant, means same phenotype. Okay. For example, here, capsule of shepherd's purse. This is the shepherd's purse plant. Okay. In shepherd's purse plant, this is the plant. This capsule, we are saying about this capsule. Here you can see. It's in triangle shape. Right? These capsules. So we are talking about that now. So if both present together, separate together, or they are present separately, that forms the same phenotype. That is the concept here. Okay. So in shepherd's purse plant, the capsule of shepherd's purse plant that occur in two forms. Okay, we have already given here that capsule occurs in two shapes. One is triangular shape and another one is ovoid shape. Okay. In triangular and ovoid shape, both shape it is present. So when a plant with triangular shape capsule, it is crossed with another which is having ovoid or top shape. Here the triangular shape capsule, a plant with triangular shape that is crossed with ovoid. This one, ovoid shape. In F1, how it will come? In F1, only triangular. According to Mendelian concept, only triangular forms will come, right? In F1, it will come in heterozygous condition and it will form triangular forms, right? So then here, these F1s, these are the F1s. The F1s are allowed for selfing. And that forms F2. Right. In F2, you can see triangular capsule and ovoid capsule. You can see triangular and ovoid. Okay. But here, see, we said already when it is coming together or when it is coming separately, it will form same effect. Okay. Here you can see both genes. Here A gene and B gene. Right. A, B, B. This also forms the triangular. Here the triangular marks are there. This is to identify which and all forming triangular forms. Okay. Here both are coming together in dominant condition. Here one is dominant, homozygous dominant, and another one heterozygous dominant. Okay. There also it forms the triangular condition. And here the next example you can see. Here first one is in recessive. First gene is recessive and second one is dominant. Then also it forms the triangular capsule. Here, check this last one. Last one, both are in recessive. Here, when it is coming together in dominant condition or separately dominant condition, both forms same result, right? Here, you can see. Here, B gene, that is in dominant condition. Still, it is giving triangular. Here, A gene is in dominant condition, but B in recessive condition. Then also it is giving triangular capsule. But here, A and B, both are in recessive condition. This one is giving ovoid capsule. Okay. So when A and B, both dominant condition, they are coming together, or A in dominant and B in recessive, or A recessive and B dominant. In this condition, all these condition gives triangular. Okay, here different combinations you can see except this homozygous recessive. Both are in homozygous recessive. Except this, all are forming triangular capsule. And when it is coming, both genes are coming in homozygous recessive condition, that forms ovoid capsule. So here, in this, among the 16, only one is forming different, like ovoid capsule. Remaining all, out of 16, 15 are triangular and one is ovoid. So here again, the Mendelian ratio. Mendelian concept. According to Mendelian concept, 9:3:3:1. Here in this Mendelian concept, this is the one and only homozygous recessive, right? So apart from this, all are forming triangular capsule, and this is the one which forms the ovoid capsule. Okay. So here in duplicate gene, the Mendelian ratio, again, that shows deviation, and it is showing a ratio of 15:1. So in duplicate genes, the ratio is 15:1. But in the previous one, we learned complementary, it is 9:7. Here in duplicate, in duplicate gene interaction, it is 15:1. Is it clear? Here triangular and ovoid, both we crossed, and in F1 that formed heterozygous condition and F1 allowed for selfing. So here what we are saying, when it is coming together or separately, when it is coming, any one allele is in dominant condition. If it is coming together or separately coming, both will give what? Both will give triangular capsules. So in Shepherd's first plant, seed capsule occurs in two shapes, triangular and ovoid shape. So ovoid shape or ovoid shaped seed capsule occurs when both genes are present in homozygous recessive condition. When both genes are present in homozygous recessive condition, that forms ovoid capsule. In all other conditions, that will give triangular capsule. Got it? Next is epistasis. So here epistasis, homozygous and recessive epistasis and dominant epistasis will come. Next non-allelic interaction is epistasis. So what is epistasis? It's nothing but the masking effect of or the hiding effect of one gene over the other gene. Masking or hiding effect. Okay. Epistasis is the masking effect. Masking or hiding effect of one gene over other gene. Okay. One gene will mask the effect of other gene. That is called as epistasis. So here genes whose effect is masked, that is called as hypostatic gene. That masked gene, that is called as hypostatic gene. So this gene, hypostatic gene effect is that won't express. Okay, this effect, that masked gene, which gene is masked, that is called as hypostatic gene. And which gene is doing that masking? Gene doing that masking effect. That is called as epistatic gene. And this epistatic gene produces that masking effect, or that gives that masking effect, and that masks the gene, that is called as hypostatic gene. Okay, here one gene or the gene pair mask or that prevents the expression of that other. other non-allelic gene. And the gene which produces that effect, that is called as epistatic gene, and that suppressed one is called as hypostatic gene. Okay. So these are of two types: dominant epistasis. Epistasis is of two types: dominant epistasis and recessive epistasis. Dominant epistasis and recessive epistasis, both are there. Here in dominant epistasis, here a dominant gene that does not allow the expression of other non-allelic gene. Okay, dominant gene will not allow the expression of another allelic gene. So this is called as dominant epistasis. In that, here you can see the dominant allele will not allow the expression of another gene. So in summer squash, that fruit color, it is governed by two genes. Fruit color is governed by two genes in summer squash. The dominant gene here, this is the dominant gene, W, capital W, the dominant gene that produces the white color. And Y dominant gene that gives yellow color. Okay. This Y dominant will give yellow color and W dominant gene will give white color. But here, when this dominant white color gene is present, that will not allow the expression of another dominant gene. Okay. So that forms the white color. So the dominant gene W, capital W, white color, suppresses the expression of the dominant gene yellow. Okay. Here you can see both genes are present in dominant condition, but here this gene, the capital W, white color gene, that is having a masking effect, that masks the effect of the yellow color gene, and that will suppresses the expression of that yellow color gene, and finally the expression of that genotype will be white. Okay. So in yellow color, when this yellow color will appear, if this yellow color appears only when this capital W, the white color gene, is absent. The W, the dominant gene, is absent, then only the yellow color will get expressed. Here, the capital W, the white color gene, dominant gene, that is epistatic gene, this one, and Y, that dominant gene, this is the hypostatic gene. Okay. So in the absence of this capital W, when it is absent, Y gene, capital Y gene, expresses. Okay. So here in the checkerboard you can see in F, see here both genes are present, that is forming white color. Here both are in recessive condition, then it is forming green color. Okay, these both are crossing together, and in F1 again it is forming a hybrid, but still hybrid, it is forming white color. Why? Here also yellow color gene is present, but the white color gene is having a masking effect over the yellow color gene. So it will produce a white color genotype or white color phenotype, not genotype, white color phenotype. Here again it will allow for selfing. So after selfing, what is happening here? Again, you can see the dominant white gene here. Wherever white dominant gene is present, all those will form white color phenotype only. Here, this is white, this one is white, this is white. Here, all, everywhere you can see white. Okay, white gene or white gene allele, any one. Here it is in heterozygous condition, but still one dominant allele is present. So it is forming a masking effect over the yellow color gene. But when we are coming to next combination, this combination, look at this. Here it is having a recessive condition. The white color gene, W gene, it is in a recessive condition, and the yellow color gene expresses here because it is in recessive condition. When it is in heterozygous dominant condition or homozygous condition, homozygous dominant condition, The Y gene expression will come. When it is in recessive condition, then only the Y gene will get a chance to express. Okay. Here again, the Y gene is in heterozygous condition. Still it is getting expressed. Why? Because the white color gene is in recessive condition. When the white color gene goes to a recessive condition, then only the yellow color gene will get a chance to get expressed, even though it is in dominant, it is a dominant gene. When the white color gene is present, that will never get a chance to express. It is having a masking effect. The white color gene is having a masking effect over the yellow color gene. And here the ratio, when the color is concerned, that trait concerned, here you will get 12 white. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All these forms white color flower and 3 yellow, because here 3 recessive white condition is coming. That's why 1, 2, 3. These 3 recessive conditions that forms yellow color. And last one, both are in recessive condition, that forms white. That forms a green. That forms green. This is a green phenotype. Okay. So this is the dominant epistasis. When it is epistasis means masking effect. Dominant epistasis means when it is coming in a dominant condition, that will create a masking effect. That is dominant epistasis. From the term you should write. Okay. Dominant epistasis means when it comes in dominant condition, then forms a masking effect. Then that won't give another gene for the expression. That won't give a chance to another gene for getting expressed. Okay. Next is recessive epistasis. So recessive epistasis is here this recessive allele, when it is in homozygous condition, recessive allele, when a recessive allele will get chance for expression when it is coming in a homozygous condition. Recessive alleles will get expression when it is in a homozygous condition. Heterozygous means again the dominant allele will get expressed. So here this is the recessive epistasis. When it is coming in recessive condition, that is having a masking effect over the dominant allele. Okay. So here example is in mice. In mice, what will happen here? In mice, wild body color, it is called as agouti. Wild body color. Okay. Wild body color. This is the wild color. This is the agouti color. This is agouti. And next, see, suppose this color, this is controlled by gene A. That is controlled by gene A. And this A is hypostatic to recessive allele C. See, when this recessive epistasis, when the recessive condition comes, the C comes together, that suppresses the other expressions. Okay. So in the presence, here dominant allele A gives agouti, we said. So here dominant C. Here, there are different conditions. Like when the C gene, when C gene comes in recessive condition, that forms what? Here dominant C. Okay, dominant C gene plus recessive A. This forms colored mice. Okay. This is the first one. Then second one, again, dominant C allele plus dominant A. dominant C plus dominant A. That forms agouti. Okay. So here, where the dominant C and recessive A, okay, recessive A, that forms colored mice. And recessive C and dominant A, that forms albino. Albino. Okay. So here when we are crossing colored one with albino, that forms in heterozygous condition, that gives agouti. When this heterozygous condition, when we are giving again for crossing, that forms these type of gametes, 4 types of gametes. And while selfing, here we can see agouti. How many are there? 9 combinations will give agouti. Then black, how many are there? 3. Out of these, 3 are black color. Okay, black color. And 4. Out of this 4 are albino, because here this is in recessive C gene, that is in recessive condition. Then it forms the albinos. When here the A gene, that is, here you can see AA. All this black color, that forms A recessive gene, that forms black color. Then agouti, how it will come? Agouti, here when it is in heterozygous condition, all those will give agouti mice. So this is the example for recessive epistasis. When it is coming in recessive condition, that forms an epistatic effect. Got it? Recessive epistasis. So we have learned different type of gene, allelic gene interaction and non-allelic gene interaction, and we have gone through different ratios. So here, which is the ratio? 9:3:4. Here these 4, when it is in recessive condition, that forms the, here another normal condition, the ratio is 9:3:3:1. These 4 forms, or it is getting changed and it is forming 9:3:4. So in recessive epistasis, the ratio is 9:3:4. You have to note down this ratio and you have to learn this example. With the help of this Punnett square, you can find out different possible combination of gametes and different gametes which are forming after each cross. Okay, that's all. Is it clear? You have any doubt? Deepa? Is it clear? You want me to repeat anything? Okay, hope it's clear. Just go through this portion. If you have any doubt, you can ask me in the next class. You have any doubt? Okay, thank you. Thank you so much.